this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
Welcome to this episode of Life Science Success. For those of you who don't know me, my name is Don, and I'm a digital marketer in life sciences. And today, my guest is Byron Purse. Byron is he is a professor as well as the co-founder of AlidaBio, and he's also a professor at San Diego State University. He's pioneering innovative approaches to studying RNA modifications. And so with that, welcome, Byron. It's great to have you on the podcast.
Well, it's great to be here, Don. Thanks for the invitation.
Yeah, thanks a lot. So could you start just by telling us a little bit about what is it that led you to life sciences?
Well, it's kind of, for me, a circuitous route. I started as an undergraduate back in the day as a chemistry or a biology and math major and drifted from that into chemistry because I realized that I had a real passion for making stuff. And I think chemistry is a really nice central science that appeals to both the tinkerer and the person who really seeks a deep understanding. And that led me to graduate work in physical organic chemistry, which is a science where you make molecules and then use these synthetic molecules to understand how molecules interact with one another and how they behave in complex systems. Eventually, from that background during my PhD, we drifted into nucleic acids when I started an independent lab at that time at the University of Denver. And, increasingly with my students, my research started to focus on how we can chemically modify nucleic acids to impart new and useful properties. And part of this was to understand some of the richness in genetic regulation, why, for instance, the same genetic code can underpin the difference in function of the more than 200 different types of cells that make up the human body. And so from that work and, doing work in academics on synthetic modifications of nucleic acids, that led me to look more kind of broadly in biology, or what are some of the natural modifications of nucleic acids beyond A, T, C, and G, or U in the case of RNA, and what are they doing? And there's a lot of really interesting unknowns in the function of all of these regulatory marks on the genetic code. So it was kind of a pathway through chemistry and creating things that led to a passion to understand, what's going on in nature at a broader level.
It is, I feel like that story of, sort of weaving back and forth or finding your path, through the path that's in front of you, is a common one as well. But I'm also really interested to hear, what is it that then led you to entrepreneurship?
Yeah, so I was a PhD student, just to kind of interweave a few aspects of those stories a little bit more, with Julius Riebeck at the Scripps Research Institute from 2000 to 2005. And in that lab, we studied molecular recognition. I studied how molecules in confinement, locked within the cavities of larger molecules, change their properties. And that's really, like, at a very kind of physical chemistry level, the science of information. What differences do molecules experience when they're confined, and how does that in a way encode information? So that kind of leads you to the connection in biology of what we're doing now, because of course the genetic code is the ultimate expression of the biological retention, use, and propagation of information. And I've always loved basic research. It's what I do in my academic life at the university, working with my students at San Diego State. There we're working on different ways that we can modify nucleic acids to make them easier to visualize, for instance, in imaging RNA in living cells, some of the areas that we're really excited about right now. But the reason I mentioned Scripps is that Scripps has always been a very entrepreneurial institution. And, even my advisor was involved in starting companies during the time of my PhD in different areas using, molecular recognition in a medicinal sense. And I met Alida's my co-founder with Alida and the company's lead and CEO Gudrun Stengel when we were together at Scripps, actually. She was working with David Miller in the same building as I was. And so you both come from that kind of background that, with a strong foundation in academics can lead you to entrepreneurialism. And I really appreciate how basic research can be a foundation for discovery and applied technologies, but how if you want to scale something up and make it accessible to a large number of people, there's a limit to how much you can collaborate as an academic lab. And for something to really hit its prime time, like a diagnostic or a widely used tool like DNA or RNA sequencing or anything in medicinal chemistry, you've got to go the commercialization route, because that's the opportunity to, develop something, make it really robust, make it scalable, make it work in everyone's hands, validate, all of these robust parameters of the product. And so that led to, my interest with Gudrun in co-founding AlidaBio, because we thought there were these really interesting opportunities to harness RNA modifications, one of these regulatory marks in genomics that I think is really hitting its stride now, but perhaps less so 5 years ago when we first founded the company on paper. And I think that you just really need easily accessible commercial tools to unravel and to apply some of these layers of biology so that, whether it's academic labs, diagnostics companies, or pharma companies, that they can take advantage of them.
Yeah, and I mean, this story of things sort of, unveiling themselves and you're finding your way, then leads you to AlidaBio. So tell me a little bit about the vision and mission of the company.
Yeah, so the idea for it came from ultimately some discussions that I was having with Gudrun, as I mentioned, our CEO and lead founder, on what we thought were some really interesting opportunities in genomics. She was working in industry at the time, had a lot of experience, in product development with companies like Illumina, Affymetrix, and Encodia, and was interested in new opportunities, not really improving existing technologies, but addressing kind of unmet needs at the frontiers of omics. So I was telling her that I was, involved a lot in the conference, like Gordon Research Conferences, the international roundtables on nucleosides and such. And there was a lot of really interesting work on these RNA modifications being done, but it was typically done by specialized labs. And, there are a lot of folks we know, and they're very good labs, but it kind of comes back to this idea of scalability. So we thought, for this whole area of epigenetics to hit its stride, it can't only be within the domain of specialized labs, no matter how excellent and, the natural limits that those groups can have to collaborate. We really need tools that are broadly accessible, that anyone is sequencing RNA could just get the sequence and also the epigenetic marks on top of it. So we thought there were a lot of opportunities there, especially as you kind of see this rise in nucleic acid-centered medicine, not just CRISPR things, but, RNA editing. There's a number of companies now looking at drugging RNA directly. A variety of technologies, of course. So we thought that the time was really right to bring to the market new tools to help this area to realize the impact that we believe it can have.
And so in terms of the overall product for Alida, then you look at yourself as a tool set provider, or can you explain a little bit more of what that would be?
Yeah, so what we wanted to do was create tools that would make it easy, not just to detect the presence of RNA modifications, but to measure the changes in those modifications. That's where biology really becomes exciting, right? I'm really into this epigenetics part of biology because, once you do genome sequencing, of a person or an organism, then you got the sequence, unless like some gradual mutations or genome rearrangement take place over time, it's more kind of like the static information repository. But epigenetics is different because it really captures what cells, tissues are doing at any given time and provides, the underpinnings for how they differentiate, like why one tissue is different from another. So we started Alida with the thought to provide the tools to make it really, easy to read, to process, to study, and use that information. But we don't want to be, we're not we're not a diagnostics company proper, but we're really excited about partnerships. So, we not only do we sell products for people who would like us to be a little bit more hands off and just want to use them, but a lot of the work that we're doing right now is with partnerships in diagnostics, pharma, and then we can really work together to understand what people's needs are, help them to make sure that they're using our tools in the most effective possible ways, and work together to make sure that this has an impact.
I think it's so important to sort of clarify that as well. And I appreciate you spending a little bit of time. And I understand how challenging some of it is as well, because, there's parts of this where you want people to use the thing that you created. But at the same time, I do feel like um, sometimes the best benefit comes through the minds that sort of, help to develop it as well. Because I've seen this in other diagnostic companies where, as they start to apply their bioinformatics pipeline to other, areas, we learn more, but also without some of the seminal knowledge that you had through the creation of, the bioinformatics pipeline, you wouldn't know, some of the things to be able to try. So it's a chicken and egg.
That's true. It all really starts with listening, and this is true of academic research too. If you get a project funded by the NIH, the number one criteria is significance, which is basically the formal way of saying who cares. Why is it going to matter? Whose research and whose interests will be impacted by your success? And with what we're doing with the company, like with any business, it starts with listening to the people who are going to use their product and addressing their needs. So, some of the things that we were hearing in those conversations was that, the quality of the data really matters. Sensitivity really matters because, since epigenetic marks underpin things like tissue differentiation, you're sometimes working with some of the most important biological samples, which could be things like biopsies. And you can't just go back to patients asking for larger and larger biopsy samples. So, working with tiny amounts of sample, getting really high quality reproducible data every time, you run the experiments. These types of things are really important. So that's why we, when we have, special opportunities to partner, we really like to, hear directly from what folks' needs are. And that helps us to make sure that as we continue to, look towards future products, that those ones really meet the needs of folks in the field, both on the industrial and academic sides.
Yeah, also important. So recently you at Alida, you launched the EpiPlex platform. So how does the technology improve the detection and qualification of RNA modifications compared to traditional methods?
Yeah, that's a great question. So, as I've already alluded to, we didn't start the field of epitranscriptomics of studying RNA modifications. Certainly not. There was a lot of pioneering work that was done at the universities, and like anything, you need to recognize when you're developing tools to detect features of biology, what are going to be the performance metrics of those tools. And a lot of the early work was done with antibodies that were raised to recognize RNA modifications. So basically what you would do is, bind modified RNA according to the modification, pull it down, separate it, and then you can sequence it. And then you infer based on what was separated that the modification was present. But those methods have had some limitations, some problems with the specificity of the antibodies, because it can be difficult to make antibodies that recognize features as small as RNA modifications. And some of those limitations have made it more difficult to get quantitative data, see how modifications change. The other thing that was really interesting to us is that there are a lot of different types of RNA modifications. A lot are in transfer RNA, but that's a very folded structure, and they're really significant there, but it's almost protein-like in some ways. And there's a subset, like 8-ish, that are prevalent in other types of RNA, like messenger RNA and long non-coding. So we wanted to be able to, read and analyze multiple types of modifications concurrently. And this got us thinking about, what's going to be quantitative, what's going to address customers' needs for small sample amounts, and how are we going to do that? One of the other early methods was chemical conversion. You basically take a modification which isn't detected by RNA sequencing. You treat it with a chemical that converts it into a mark that is picked up on the sequencing. But my background is in chemistry. It's pretty challenging to develop reactions that operate on an entire pool of RNA and cause no damage, only impart the one effect. And that problem becomes exponentially more difficult if you want to do multiple types of modifications altogether. So this was kind of like the birth of the idea of EpiPlex. The idea was we would recognize the RNA modifications by binding. The advantage there is that you don't have to deal with these challenges of orthogonal complementary chemical reactions that can all work in the same environment, and they're non-degradative. And then not only would we bind and enrich, but we would also barcode. And what that means is effectively genetically recode the presence of the modification by adding an artificial sequence of DNA to the target that artificial sequence, like a product barcode at a store, provides information. And that's the identity of the modification. So EpiPlex binds modified RNA, transfers a barcode to it, and then once that RNA is sequenced, allows the reading of the barcode to infer or to remember that the modification was present.
So then I'm assuming then that you then are sequencing to try and target those things that are barcoded on the back end?
Yeah. So, people are really interested in knowing too, just, what's the RNA expression profile, which RNAs are present in their system. So we split the sample so that, intact, basically non-epiplexed RNA passes through, and we use that as a control. So people can see the entire pool of natural RNA in a sample. And then the barcoded parts are specifically modified fragments. And with EpiPlex, we built an entire bioinformatics suite called EpiScout, which is accompanied with the product. And that makes it easy for, I'll say non-expert users. They are expert users, but maybe they don't want to dig deep into coding in bioinformatics to use their data. So people who would be doing, DNA or RNA sequencing anyway. So that makes it easy to, access and to see, where are the modifications, how they change between different biological conditions and such.
So how do you see the role of RNA modifications or the epitranscriptome impacting in future research and clinical applications, particularly in terms of disease diagnostics or therapeutics?
Yeah, I think there's a lot of ways to answer that question because in some ways it's early-ish days for the field. As I'm explaining, our motivation was, building tools for accessibility so that anyone engaged in RNA research can study these regulatory features. So I think first there's a lot of more to be done in basic research, to understand how RNA modifications are dysregulated in disease, how they may play roles in pathogen-host interactions, how they may be associated with aging, developmental disorders and such, because, understanding connections between genotype, epigenotype, and phenotype is where the whole process of diagnostics and medicine starts. So there's that angle, and I think there's a lot of opportunity there. From the diagnostics perspective, because RNA modifications are frequently dysregulated in disease, and sometimes they're drivers of disease progression because they can be involved in things like the inflammatory response, the immune response, and such. We have found through some of the studies that we've done internally and with partners that if you look at diseased versus normal samples, these can be tissues, these can be blood samples, that you can perform what's called PCA, principal component analysis, a kind of a broad data analytic method for clustering. And you can see differences between health and disease in things like just the RNA expression levels, but you can get much better clustering if you include RNA modifications because now you've added all of this richness. So we think that there's a lot of untapped opportunity directly in biomarkers within our RNA modifications because they can correlate with disease, even if those modifications are not measured just in the diseased cells themselves. For instance, you might be able to pick that up in the immune system. And therapeutically, there's a number of different opportunities there. One thing that's excited us since the early days is that there's a UK-based company, Storm Therapeutics, that has recognized that dysregulation of RNA modifications is really important in enabling cell proliferation and functions as a driver in cancer. So they are actually directly drugging RNA modifying machinery and seeing they've published some clinical trial results that are publicly available and shows some really exciting potency and some ability to slow or even reverse tumor progression. So of course if you're drugging pathways that are involved in RNA modifications, then measuring those types of things, have the potential to be valuable. And there are a number of companies that are looking at doing things like modifying RNA in cells, like A to I editing or RNA silencing or RNA interference. So anytime that a therapeutics company might like to understand the targets they could be engaging in RNA-centered medicine, we can reveal more complete information on the nature of the target by revealing not just the sequence of the target, but its modifications. So I think these opportunities span the entire range of the things that you've mentioned.
Yeah. I mean, and at least for somebody that isn't a chemist or doesn't come from this world, from my perspective, it's exciting to hear as well. So you've talked a lot about the partnerships that you've had. Let's talk a little bit about your partnership with DNA Nexus to enhance RNA research tools. How's this collaboration advanced precision medicine and personalized treatment approaches?
Yeah, so DNA Nexus, for those who don't know, is a cloud provider where you can store genomic information and work with tools to analyze it. So, I've mentioned several times that a big part of our focus is on the accessibility of these tools. So, genomic data involves pretty substantially sized data sets. So for those who are, running our assays, for instance, with the kits, they can store their sequencing data within the DNA Nexus cloud and analyze it with our EpiScout software directly within that cloud. So it makes it a lot easier because folks who are interested in or experienced with RNA sequencing can literally then get push button analysis within that platform where they'll be able to access, or they can access now, not just be able to, our reporting tools that show, for instance, which sites of the transcriptome, the totality of RNA, are the most differently modified between different biological conditions. So it's just there to kind of like turbocharge discovery, to help people to pick out that significance in a quick and accessible way. And we also offer, a full download of all of the sequencing data for expert users. So those who want to dig deep, do a little bit more coding and such, they have that opportunity available.
Wow. Can you share a particularly challenging or aspect of developing the EpiPlex platform, and then how did your team overcome it?
Yeah, so there are a few different things I'd like to comment on there. I think I'll comment on two. So, I mentioned that we use binding agents to recognize RNA modifications. And I mentioned that the field began with antibodies, and we've tested a bunch of them, and some of the different properties of antibodies are also discussed in the peer-reviewed published literature, and they can be a little bit hit or miss. And I think the challenge is, if you think about what the immune system does and how it can be used artificially against unnatural targets, then you can kind of recognize that there's a problem. The immune system is programmed, the adaptive immune system, to recognize foreign materials, but it doesn't have a great counter selection method, which means it can develop antibodies that bind something with pretty high affinity, but there's no way to train it at the same time to make sure it doesn't bind something similar that's just a little bit different, right? And that's actually really important in this context because RNA modifications tend to be small, and we need to make sure that the binding agents don't just bind to DNA or RNA in general, which the antibodies do to a much greater extent. So to make the assay work well, what we did was we turned to protein engineering, and we took broadly from the family of RNA binding proteins. I can't say specifically which ones, but there are a lot of them that exist in nature. and then used modern protein engineering methods, which allow you to have both positive selection and negative selection. So we can make sure that they bind specifically to the modification of intent and that they don't have a lot of affinity for either other modifications or just unmodified DNA or RNA. And that has really been very helpful with, optimizing assay performance. And, the other thing that you can do with these artificial methods is you can control how quickly things bind and are released. And since our assay uses beads, making sure that they have similar rates and they play well together really helps to give the assay excellent performance. The other thing that's really important, and I think one of the biggest challenges in the entire field, is that because it's early-ish times for RNA modifications, there isn't a really widely agreed gold standard, like a truth set for the field, and that's one of the biggest challenges. So what we've done is take two approaches to that. Some of this stuff is in literature that we published and is accessible on our website. We've compared our methods with a variety of some of the most widely used things in the academic literature to kind of look at overlap. So that information is available. And the other thing we've done is we've synthetically made completely unmodified RNA and then rerun the assays with that. So basically, you just create RNA using transcription in the lab. And you can check, because if you know that it's unmodified, so it shouldn't give signals in the assay. And we have a very low false positive rate from those experiments. The other things we've done is to do things like take genetic manipulation technologies like knockdown or knockout, and you can deplete cells of their ability to install RNA modifications and then check to make sure that the assay reports a loss of modifications that matches the biology. So between all of those things, I think we're really confident that EpiPlex is delivering truthful information. The reproducibility is definitely great, and I think we're aligned with the standard of the art as the entire field moves towards agreeing on what the gold standard is.
Mm. I appreciate you sharing that, because I one of the things that as I was developing the podcast, I remember reaching out to several people saying, "Hey, look, I have this podcast called Life Science Success," and they said, if you find success in life sciences, then you're really not working hard enough. That there's a lot of failure that happens in this field, and I'm like, yeah, but at the same time, the thing that makes it successful, I feel like, are the fact that not only do we have a challenge, but then we find our way through. And then on the other side, there's, not just the science, but the understanding of, hey, look, here's what sort of moved that progress forward. So anytime I get to these questions and I have somebody sort of unveil, a little bit more of the story, I really appreciate it. So thank you.
Well, I mean, I think innovative research is always difficult research, because if you're pushing the frontiers of science, it doesn't matter whether this is at industry or at a university, you are by definition pushing up against the frontiers of the unknown. And all of the training that you have, and like the wisdom and the great ideas that come from your team are really key to try to make the best possible decisions. Pick which path offers you the highest chance for success. Think physical chemistry principles on what's reasonable and what's not, and then make a really targeted search for the best type of opportunity. And certainly, like, innovation and creativity in the ways that you can solve problems are absolutely key. But yeah, developing new technologies, no matter what you're doing, is definitely difficult. There's no way around that.
That's for sure.
Mm.
What's the greatest piece of leadership advice you've ever received?
I have a few things to say there. I think maybe one of the most important ones is that respect is earned and not given. I mean, treating other humans beings with a basic standard of respect, I think, is kind of normal. But I think as a leader, you really have to lead by example, and show the dedication to quality, the openness, the inclusiveness, the commitment to innovation and excellence that you expect of your people. Because if you're not exemplifying that yourself, I think it's difficult to expect it from others. And I think the other thing that's really important is to recognize that great ideas can come from anyone and anywhere. And great leaders will really harness, I think, the intelligence and the creativity of their entire team, no matter how junior, and then still be transparent with responsibility. Because, sometimes you'll have to make a strategic decision or a business decision that will be made with imperfect information and may not match the opinion of everyone. But as leader, you got to take ownership for it, right? You get the credit if it's a great decision, and you're responsible if it isn't. And I think having an inclusive environment where you can listen to great ideas from even junior team members, sometimes they're terrific and you want to run with them. But if you make a different decision, just having the openness and transparency to say, I really appreciate that idea. I like the way you're thinking. We're going to do something a little different, and here's why, I think is a really important part of leadership, because it keeps all of those good ideas flowing. And the most successful leaders draw on all of these different sources of inspiration and ideas and use those to pick the best path forward.
I think it's an important thing to sort of help people understand what your decision-making process was too, just from the standpoint of, like you said, it's never very clear as a leader exactly, which direction you should choose. You have a limited set of data that you have to make a judgment call on at the end of the day. And that judgment calls could be based on your own personal history, could be based on, hey, look, going this way routinely has worked for me. I'm going to go that way. However, it also could be, maybe the unique thoughts that are being posed are ones that challenge your level of thinking as well. And so I, as a leader, I know I appreciate all aspects of it as well.
Yeah, I think another thing that's important is that one should always reflect on, how reversible a decision is, because indecision, I think in any organization can lead to paralysis. Some decisions are thankfully easy. Some are really tough. And if you head down the wrong path, actually not as bad as they might seem on day one to actually reverse and change direction. So it can, even if information is perfect and there's some uncertainty in decision making, I think it's often better to pick a path, use the best judgment that you can. And if it's not that hard to pivot, if you realize it's not right, that's a better strategy than just waiting and not deciding.
No, also true. Byron, what inspires you?
Opportunity and the unknown, I think. Challenge too. I mean, I really enjoy the way in research the path to success may not be entirely clear. The fact that there are going to be challenges, and correspondingly the great feeling that you can get when you really make something work. It's, you just draw kind of a weird analogy, like if you ever play video games on invincibility mode and then beat the boss. I mean, it's just not that exciting, right? But if it's going to be difficult from day one and then you find the success there, it's a tremendous feeling. So I think working in challenging and important areas and striving hard for that success can give such a tremendous reward when you really manage to make an important innovation when it wasn't easy from day one. That inspires me. And then kind of thinking what the next opportunity will be. Some advice so one thing that really resonated with me a few years ago is that my PhD advisor Julius Riebeck was asked in an interview what he saw himself doing in 5 years, and he answered, "If I knew that, it wouldn't be worth doing it." I thought it was such a cool answer, because it's a reflection of how, science and the problems that we face can change actually really rapidly. And it's important as a scientist, in both academics and in industry, to think always about, what's the significance of the problem that you're trying to address. And significance can change over time. So what's an important problem this year may look a little different a few years down the road. And I think looking ahead, it's always great to have ideas and to look ahead, but one should be aware that the future can change a lot.
I remember this from my PhD studies. It was my favorite part of research papers became that end section where the researcher would essentially say, these are other unanswered questions that I'm leaving on the table. And it was one of those things where I sort of, it was a great mystery that I sort of uncovered through reading all of these papers, that, some of the greatest questions that you could ask next were always at the end. And I loved that aspect of it. And to me, that's what I hear in your answer as well, is that, look, I mean, sometimes you just don't know what those questions are going to be, what's going to be still left on the table as you get to the end. But you can also, use that to leverage it to the next opportunity as well.
Yeah.
Byron, what concerns you?
That's a tough question to answer in this climate. I think the main concern I have right now is kind of like it's almost like a political thing, but I don't really want to make this too political. I think there are too many headwinds right now that are too protectionist in focus. What I mean is that, the global research enterprise has always been a competitive one. Countries always try to, be the top dog, the leader in different technical areas. And I think it's important to recognize that it's not a zero-sum game. Companies definitely, or countries definitely benefit from being on top, but all of us, all of humanity benefit from a global push, to advance technologies, with better solutions, better opportunities for human health, cleaner technologies, cleaner energy sources, and things like this. So I think the headwinds right now have a little bit too much focus on protectionism rather than investing and competing at the higher level. And my concern is that, the global, certainly the national research climate over the next years will focus a little bit too much on barriers and protectionism and not enough on investment, openness, and excellence. So hopefully we see some movement in what I think would be a more productive direction.
I mean, I'll take a bit of an aside in that, and would love to sort of test this thought with you. So, I mean, my greatest fear is in 10 years, somebody turns around expecting the next round of whatever drug, whatever diagnostic or whatever, and there's a massive gap that all of a sudden everybody recognizes what, limiting NIH funding has done and what, some of the challenges that many of the companies have faced into right now. I think there's a couple of different routes that you could go, but mentally, my one thought is, it would be remiss if also the corporations that were out there that know that they need technologies didn't also adjust for the climate that we're currently in. And so, I think there is an opportunity for corporations to say, hey, look, how can we sort of not let this impact us in the future and sort of pick up on things that we think, would really benefit our company. But it also means that their investment profiles have to change. And if they don't change and they stay the same, unfortunately, this void or the valley of death widening that I'm sort of, drawing a picture of is that, there will be a gap. And unfortunately, people will, I have a feeling, turn around and see all the things that didn't make it through the valley of death during this period of time. But, I don't know if that resonates with what you're saying or not, but.
I mean, I'm an optimist by nature. And I think that, certainly, in California, I think, broadly, within the American research community, there's a lot of scrappiness, there's a lot of spirit of innovation, and there's a lot of willingness to, that Silicon Valley mantra, fail early, fail well, dust yourself off, and quickly pivot and do something different. So I think there are a lot of very strong kind of innovative culture things that really, help us to be kind of in a strong position. And, as I've said before, the global research enterprise is not a zero-sum game. So I think there's a lot of great discovery, a lot of beneficial things coming from everywhere. So I think we'll see what happens. I just hope, I think we should be spending a little bit more, we as a society, on R&D.
Yes. Last question. What excites you?
The future. There are so many changes that I think we'll see, as for instance, in human health, right? As we understand new diseases through innovations in epigenetics, what AlidaBio is trying to do, as we see this revolution in medicine, programmable medicines based on nucleic acids, right? I mean, you can consider these RNA vaccines that emerged into popular use during COVID. But that wasn't a technology that was created because of the potential for a viral pandemic. It was created to do things like create personalized immunotherapies for severe forms of cancer. And, the magic is really that the building blocks are all the same molecule and it gets degraded all into the same byproduct. So these types of technologies, or CRISPR, for instance, right? Editing in its derivative technologies, through companies like what Beam Therapeutics are doing, these incredible capacities to correct genetic problems in a huge amount of genetic information. The genome's 3 billion base pairs, right? To do precision edits is like a miracle of chemistry, science, and biotechnology. So I think, as we see new paradigms in medicine, along with a lot of innovation, honestly, in the small molecule portfolios and drug discovery, new paradigms in epigenetics and their ability to inform precision care and diagnostics. I'm pretty optimistic that we will see in the next 10 to 20 years massive strides in personalized medicine and human health. So, a lot people were talking this up 20 years ago when the Human Genome Project was published. But I think people always talk up stuff a little bit too much, and then the revolution comes, but it's a little slower than what people have in mind. You can see over the past decades enormous strides within cancer treatment across many different cancer types. Right? It wasn't that from one day to the next there's a cure for cancer, but over decades, massive improvements. And I think between epigenetics, and we hope that AlidaBio will play a part in this, precision medicine and new approaches to identify small drugs and therapeutics will have transformative advances in the ability to understand human health, to detect the emergence of disease early on, and to take more effective action.
Yeah, the biggest part of the hype curve that I remember overall was my start in life sciences. I remember one of the scientists I was working with, he had said he was so excited at that point in time about monoclonal antibodies. A decade later, we start to see like the first monoclonal antibodies having their biggest impact on humans. And so, it was, to me, it was one of those points where I was like, I remember talking about this thing way back whenever, standing in a lab learning about bioprocess, and then essentially, now all of a sudden, it's becoming a reality. So, for sure, we do see that, but at the same time, with all the good people and all the scientists, plus all the research, I kind of go back to my thoughts on COVID, really were in working with a couple of different companies through that time period, and then listening to them talk about their previous research, with SARS and other, sort of global, pandemic possibilities, and then saying, hey, look, I think these things have scientific ties. And again, all of a sudden, out comes an opportunity to at least vaccinate for COVID as well. Mm-hmm. Well, Byron Purse, thank you so much for being on the Life Science Success podcast, and thank you for telling us about AlidaBio. It was a great conversation. I appreciate your time and for just the conversation about what you guys are doing and the impact you're having.
Thanks so much for the opportunity, Don. I really enjoyed this.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it, and last but not least, rate the podcast. Thank you again.